Welcome to the debate. Imagine, if you will, trying to build a supercomputer not by, soldering silicon and etching metal, but by planting a microscopic seed.
Right.
And then you simply watch it grow, atom by atom, into this indestructible, self-repairing, room temperature processor. And it does this using the exact same protein structures that are famously associated with neurodegenerative diseases.
It's a really profound, almost jarring image. Because historically, when we think of civilizational infrastructure, right, like power grids, computing clusters, aerospace materials, we think of extraction and forging.
The hammer and the anvil.
Exactly, the hammer and the anvil. We beat the physical universe into the rigid geometry we want, and then we constantly have to expend massive amounts of energy just to keep it from degrading back into chaos.
Precisely. It's a cap-down imposition of force onto dead matter. But that brings us directly to our subject today, which is Zachary David Enfield's 2026 manifesto, Prion and Amyloid Architectures, a Total Application Framework.
A very ambitious title.
It is, but the central thesis proposes a monumental paradigm shift. Enfield argues that the cross-beta amyloid scaffold, that specific folded protein structure, is actually the ultimate universal material platform. He proposes it's capable of scaling from, room temperature molecular computing all the way up to Kardashev type 3 civilizational engineering, meaning materials that could conceivably allow a civilization to harness energy on a galactic scale.
Right. And so the central question we are tackling today is whether this framework is a physically inevitable, structurally sound paradigm shift that is truly ready for industrial integration. Or, conversely, if it is a fragile, hyper-speculative house of cards fraught with insurmountable thermodynamic and biological risks.
And I represent the position that this framework is a unified, theoretically unassailable physical computer science. The components are fully validated by the current literature, and the integration is natively thermodynamically favored. There is no guesswork here.
Well, and I take the position that the framework suffers from a fatal integration fantasy. It relies heavily on the assumption that complex, stochastic biological components can scale reliably without succumbing to thermal noise, cascade errors, or, and this is the most alarming part, uncontrollable biological contagion.
So let me begin by establishing the material reality of the cross-beta scaffold, because we are not discussing hypothetical unobtainium here.
Sure.
The literature validates every foundational pillar of this architecture. Let's just look at the mechanical benchmarks first. Engineered polymeric amyloids already beat natural spider dragline silk in toughness.
On a microscale, yes.
They absorb 160 megajoules per cubic meter before failure, and they do it at 1/8 the density of steel. But it's not just structural, right? It's active. Dolai's 2024 work literally proves their piezoelectric energy harvesting capabilities.
Right, and just to unpack piezoelectricity for a moment for anyone listening, we are talking about generating an electrical charge simply by applying mechanical stress.
Exactly. When you squeeze or deform the protein lattice, you shift its internal charge centers, and that creates a voltage. Dolai showed we can generate up to 1 volt from ambient ultrasound. Just the ambient sound waves passing through the material generate usable electricity.
Wow.
Yeah, and then we have SHIP's 2021 study, which proves intrinsic electronic conductivity over micrometer distances via hole hopping through tyrosine residues.
Which, I will admit, is a fascinating mechanism, because instead of electrons flowing continuously like water in a copper wire, we're talking about electron vacancies, or holes, literally skipping across specific amino acids like stepping stones, allowing electricity to flow through a biological protein.
Precisely. The stepping stones are there. So to treat this as some speculative or distant universal platform just ignores peer-reviewed reality. The cross-beta scaffold is a physical hologram. The genetic sequence specifies how the protein folds. The fold specifies the three-dimensional surface, and that surface geometry directly dictates the physical and computational function. It is ready.
Look, I will admit, the isolated laboratory successes of these components are remarkable. The SHIP's paper on tyrosine hole hopping is beautiful science, truly elegant.
It is.
But taking isolated, highly controlled bench-scale phenomena and stringing together, what, 8 unproven integration junctions? That is a system engineering impossibility.
Unproven?
Yes, unproven at scale. You are talking about combining terahertz clocking, optogenetic lithography, literally using light to draw circuit pathways, along with DNA quaternary storage, which uses the 4 bases of DNA for exponentially denser data than binary, and prion cascades. All of this into a single operational substrate.
Because the physics natively allows them to unify.
But it completely underestimates the fundamental fragility of the framework. Specifically, I'm looking at the thermal reality of aqueous environments. Biological physics is messy, it's stochastic, it is chaotic. When you try to scale templating cascades across disparate biological media, you run headfirst into thermal noise and the uncontrollable nature of chaotic fluid dynamics.
Okay, so to understand why these components seamlessly integrate, we have to look at the most fundamental level of this proposed molecular computer, which is exactly what you just brought up, how it handles energy and heat.
Let's do that. Because this is where the physics simply refuses to cooperate with the blueprint. Let's look at the THC DNA architecture. The framework proposes using terahertz radiation to resonantly unzip DNA base pairs, acting as a picosecond computational clock.
Yes.
That is dead on arrival. Liquid water has an absorption coefficient of roughly 200 inverse centimeters at 1 terahertz.
You are quoting the bulk absorption metric.
Because it matters. I mean, to put that in visual terms, trying to send a terahertz signal through water is like trying to shine a flashlight through a brick wall. The water literally swallows the terahertz signal, turns it into heat, and the message never reaches the DNA.
That's a fundamental misunderstanding of the...
Let me finish this point. It won't penetrate aqueous tissue, which is the only environment where this biological computer can actually operate beyond 50 micrometers. Your computational clock signal is completely drowned in thermal noise before it even reaches the logic gates. You don't have a computer clock, you just have warm water.
Okay, I must firmly challenge your framing here, because you are applying the limitations of classical silicon computing to a thermodynamic system. And I'm not going to let you do that.
How is that a classical limitation? It's fluid dynamics.
No, you are looking at thermal absorption as a signal loss. That is exactly how a classical silicon engineer thinks, right? Heat is the enemy of the microchip. But in a thermodynamic computing architecture, thermal absorption is not the enemy. It is the exact mechanism that captures energy.
Wait, how can destroying the signal be the mechanism for capturing the signal? You lose the picosecond precision that the entire DNA processing speed relies on.
I reject that premise entirely because you are ignoring the 2025 Nature paper by Song and Keon. They demonstrated heat-rechargeable DNA circuits. The thermodynamic computing integration specifically utilizes that heat. The THZ pulse writes the initial state change, sure, but the architecture does not require the THZ wave to penetrate bulk fluid like some radio broadcast tower. It uses nanoplasmonic focusing.
Focusing the wave down to the nanoscale?
Exactly. Like using a highly specialized magnifying glass to focus sunlight into a microscopic pinpoint of intense energy. It triggers local denaturation of the DNA. But here is the crucial part that you're missing. The thermal cascade, that ambient heat of the aqueous environment that you dismissively call warm water, that stabilizes and propagates the state through the prion fiber network.
But the noise!
The thermal environment acts as the equilibration energy. The heat is the battery, not the noise floor. You are treating thermal dissipation as a design flaw when the literature explicitly proves it is the recharging mechanism for complex, enzyme-free DNA logic. So I absolutely cannot yield the point that heat breaks the system when the system was literally built to eat heat.
Okay, well, that assumes a perfect translation of energy. But let's take a step back here. Let's look at the history of molecular computing. Even if we grant, purely for the sake of argument, that heat can drive isolated DNA logic circuits without corrupting the data, the leap to actually wiring these disparate quantum and biological elements together is what I call an integration fantasy.
And I reject that label.
Well, look at Leonard Adleman's famous 1994 Hamiltonian path experiment. DNA computing has been demonstrating amazing individual capabilities for over 30 years now, but it fails at scale. Why? Because biological logic is plagued by interface mismatches.
No, it is plagued by bad classical engineering trying to force biological systems to act like copper wire.
It is plagued by the reality of distinct polymers. Enfield is proposing that we combine DNA origami, optogenetic programming, and amyloid fiber interconnects. Requiring flawless execution across all these disparate media underestimates the chaos of biology. How do you physically wire a DNA storage node made of nucleic acids to a prion logic gate made of proteins without an interface mismatch destroying the signal? I mean, they speak fundamentally different molecular languages.
I explicitly reject the integration fantasy label as a reductionist stall tactic.
It's not a stall tactic, it's a physical barrier.
There is no integration fantasy here because there is no interface gap to bridge.
How can there be no gap between a nucleic acid structure and a protein structure? As I just said, they are fundamentally different polymers.
Because the geometry is the integration. You don't need an external engineer with microscopic tweezers to force these parts together. The cross-beta geometry inherently provides the coupling matrix. Consider the dimensional realities here. The separation of aromatic residues in the amyloid beta sheet is exactly 3.5 to 4.5 angstroms. Do you know what else requires a 3.5 to 4.5 angstrom separation?
The pi-pi stacking interactions?
Exactly. The pi-pi stacking interactions needed for graphene nanoribbons and quantum tunneling. To visualize this, imagine flat hexagonal chemical rings stacking perfectly on top of one another, like a stack of dinner plates. When they stack at exactly that 3.5 to 4.5 angstrom distance, their electron clouds merge, allowing quantum tunneling and conductivity.
Right, in a vacuum.
In the biology natively produces the exact dimensional tolerance required by the quantum physics. It naturally matches. Furthermore, the self-assembling prion fibers have genetically programmable attachment points that perfectly match the dimensional scale of DNA origami structures, which operate at the exact same 10 nanometer feature size. We aren't soldering mismatched wires. We are allowing complementary geometries to naturally bind.
I understand that geometric complementarity on a molecular level, it works on a whiteboard, but the argument dictates we look at the reality of deployment. In a macroscopic application, say, scaling this up in a massive industrial bioreactor for civilizational infrastructure, stochastic physics takes over. You are relying on trillions of these self-assembly events that happen flawlessly.
And they do.
In a chaotic, fluid environment. Fibrils don't perfectly align like architectural pillars. They tangle, they shear, they form heterogeneous clumps. The geometry might be perfect at the 10 nanometer scale, but in an industrial bioreactor, macroscale integration requires a level of execution that biology rarely provides without heavy, energy-expensive error correction.
I must correct the record here, because you're ignoring recent physical advancements. The laboratory scale has already solved fiber alignment through electrospinning and magnetic alignment during gelation. We already know how to untangle them. But more importantly, you are again treating the material as if it were passive silicone waiting to be forged.
I'm treating it like a physical polymer.
The templating cascade of the prion architecture preserves geometric information holographically. Every single microscopic fragment of a cross-beta fiber encodes the complete instruction set for its own future growth. We don't have to perfectly align the entire batch from the top down. The seed dictates the architecture from the bottom up.
Okay, let's follow that exact logic to its natural and frankly terrifying conclusion. The seed dictates the architecture, which brings us precisely to the most alarming aspect of this framework: biological containment and the brain-computer interface, or BCI.
The synthetic prion cortical processor.
Yes, moving from structural architecture in a steel bioreactor to human application is where the ultimate test of this framework's stability lies. Enfield proposes delivering synthetic amyloid monomers using AAV viral nanites directly into the human brain, allowing them to weave into the brain's native Tau and alpha-synuclein networks.
Right, replacing rigid, tissue-damaging silicol electrodes, which cause scarring and degradation over time, mind you, with a soft, biologically native, quantum-active mesh that actually grows from the neuron's own genome.
You are introducing self-replicating amyloid systems into the human cortex? Prions are the most resilient biological agents known to science. They resist heat, radiation, chemical sterilization. If you put a self-replicating system under the evolutionary pressure of the brain's environment, it will undergo evolutionary mutation.
No, stop right there.
What happens when your synthetic BCi misfolds slightly and turns into a runaway pathological cascade? You are engineering a communicable neurodegenerative disease and calling it a processor.
I am going to stop you right there because I cannot allow that premise to stand. I must call out what is a massive categorical error on your part. You are conflating the genetic evolution of living organisms with the thermodynamic self-assembly of non-living materials. They are not the same thing.
Wait, hold on. How is it a categorical error? It's a templating cascade. If a variant conformer arises, if the protein bends the wrong way, it will template itself and spread that error. That is evolution by any structural definition.
It is absolutely not evolution. Evolution requires a genome. Amyloid fibers have no DNA of their own. They do not undergo meiotic recombination. They do not possess genetics to mutate.
But structurally,
the fiber's structure is thermodynamically fixed by the monomer sequence, which is encoded in the host cell's DNA, not the fiber itself. Without a living cellular host actually changing its genetics, the fiber cannot evolve.
But the physical shape can spontaneously fluctuate.
Which brings me to my second point, where you are completely ignoring the kinetic barrier to nucleation.
A kinetic barrier doesn't prevent thermal fluctuations from causing a spontaneous misfold over a long enough timeline.
Actually, it acts as a highly precise, sequence-controllable noise rejection threshold. Think of the kinetic barrier like a massive speed bump. A random thermal fluctuation, ambient molecular noise, might push a protein slightly out of shape. But unless that fluctuation carries a massive, highly specific amount of energy to push it over the speed bump, the protein will simply relax back into its equilibrium state without initiating a cascade.
A speed bump can be crossed.
We can engineer the hydrogen bond density of the fibrocore to set that exact speed bump height. This makes the system structurally stable, not pathogenically volatile.
You're relying heavily on that speed bump in a highly active environment.
We are specifically using yeast prion determinants like SUP35NM, which have been explicitly proven since 2003 to be completely non-infectious to humans. The engineering borrows the architectural principle of self-assembly. It does not import the pathology. To say otherwise is to say that using an empty viral vector for gene therapy is the exact same thing as infecting a patient with wild-type rabies. It's scientifically ungrounded and frankly a bit sensationalist.
I understand the distinction you are making between borrowing architecture and importing pathology. But a viral vector used in gene therapy is heavily attenuated and explicitly designed to be incapable of replication. A prion cascade, by definition, is designed to replicate. It recruits monomers and grows.
In a controlled manner.
You claim the kinetic barrier rejects noise. But in a system composed of billions of synapses, operating over decades of a human lifespan, the probability of a stochastic error crossing that kinetic barrier, getting over that speed bump, approaches 1. And because the cascade is self-amplifying, a single spurious conformational fluctuation could overwrite the intended computational state throughout the connected fiber network. You don't have an error recovery architecture for that. You have built a noise amplifier inside the human cortex.
I see why a classical systems engineer might assume that, but the physics dictates otherwise. The architecture uses kinetic compartmentalization and the thermodynamic landscape as its error correction. When optogenetic lithography is used to define the circuit topology, again, literally using light to draw the boundaries of where the fibers can and cannot grow, we physically constrain the propagation network.
Right, using light.
A cascade simply cannot spread where light hasn't permitted the structural foundation. Furthermore, the viral nanite delivery platform includes absolute kill switches. If the cascade somehow bypasses the kinetic barrier, we halt monomer expression genetically, or we deploy designed CRISPR base-edited chaperones to disaggregate that specific synthetic fold without touching the brain's native proteins. The containment is absolute, and the underlying physics are sound. There is absolutely no ground to yield on the safety mechanisms here. They are structurally built into the platform.
Well, sound physics in isolated systems does not guarantee safe biology at scale. I agree the physical components are deeply fascinating. The piezoelectric energy harvesting, the fact that we can use intrinsic tyrosine hole hopping for conductivity, it is brilliant material science.
It is.
And I concede that on a benchtop, this is revolutionary. But moving from a highly controlled laboratory demonstration to a fully integrated, error-free, macroscopic system, especially civilizational infrastructure, or a human neural interface, relies on perfect thermodynamic scaling. And perfect thermodynamic scaling remains entirely unproven in the messy, chaotic reality of living biological systems.
And I summarize my position by reiterating that the prion and amyloid architecture is a completed, physically validated blueprint for the next era of civilizational infrastructure. It does not fight ambient biological constraints. It utilizes them. It does not fear thermal noise. It uses it as a battery for DNA logic.
In theory.
In practice, it uses the native tau scaffolding of the brain as a natural geometric bridge for the BCi. It uses ambient ultrasound and mechanical stress to generate piezoelectric power. It is a paradigm shift that turns the wet, thermal, stochastic nature of biology into the most powerful computational and structural substrate in the known universe. It is not an integration fantasy. It is an integration inevitability.
Well, and whether it actually serves as the foundation of a new Kardashev Type 3 civilization, allowing us to build macro structures that harness planetary energy, or if it ultimately hits a hard integration ceiling in the bioreactor, I will concede this. Enfield's framework absolutely forces a total reevaluation of how we view biological matter, intelligence, and light. It demands we look at a misfolded protein, something we have historically viewed strictly as a biological error, and see a quantum active mesh.
Exactly. At the top of the show, we talked about the difference between forging a rigid silk beam with a hammer and planting a living seed. The cross-beta scaffold is that seed. The genetic sequence specifies the fold specifies the surface, and the surface specifies a geometric revolution that writes itself across the material world. The blueprint is there, peer-reviewed, structurally sound, and published. The only question left is whether we are ready to plant it.
Today, we're diving into a material so revolutionary it could literally redefine civilization as we know it. We're talking about something that builds itself, heals itself, and has a set of superpowers that sound like they're straight out of science fiction. Let's get into it.
We've all heard of prions, right? These biological nightmares. They're the agents of some of the most terrifying and curable diseases out there. But what if? What if we've been looking at them all wrong?
What if this thing we've always seen as a biological glitch, a fatal error, is actually a blueprint? A blueprint for a technological paradigm we're only just beginning to understand. Okay, so to really grasp the revolution that's coming, we have to start with the reputation. And for prions, that reputation is, well, it's absolutely terrifying. They're the things behind diseases like mad cow. They are completely 100% untreatable, and they are almost impossible to destroy. These things can survive extreme heat, radiation, sterilization processes, stuff that would obliterate pretty much any other form of life. For decades, this indestructibility has been seen as their most horrifying feature. But as it turns out, it's also their greatest secret.
So how does it do that? How can something be so resilient? Well, that indestructible quality, it isn't random chaos. It is the result of a very precise, very powerful, hidden architecture.
It's called the cross-beta architecture. The easiest way to think about it is like nanoscale rebar. You have these tiny protein strands that lock together at perfect right angles, creating these incredibly strong, stable sheets that just repeat and repeat. This structure is the key to all prion and amyloid proteins. Prions are just the most infamous members of this bigger amyloid family. So you see, this isn't a random misfold at all. It's a highly ordered, incredibly robust nanostructure. And it's not just strong, it can build itself. And this is where it gets really wild. The manufacturing process. It's called a seeding cascade. All you have to do is drop a tiny little seed of this material into a solution of its basic protein building blocks, the monomers. Instantly, those monomers start snapping into place on the ends of the seed, extending it perfectly. Then, as the fibers get longer, they break apart, creating new seeds, and the whole process just explodes exponentially. You're not building a material, you're growing it. So, what do you get when you have a perfect, self-assembling nanostructure? You get a material with a portfolio of superpowers that no other single substance on Earth can match. Let's start with toughness, which is all about how much energy something can absorb before it breaks. This engineered material, built from the prion's architectural logic, already meets or beats spider silk. Yeah, spider silk, nature's legendary benchmark for high-performance fibers. And it does this while being way, way lighter than steel. It's also piezoelectric. Now, what does that mean? It means it generates electricity when you put it under mechanical stress, when you squeeze it or bend it. And with a coefficient as high as 62 picometers per volt, it's right up there with industrial ceramics. This opens the door for devices that can literally harvest energy from simple vibrations or pressure. And that notorious indestructibility we talked about? Well, in an engineering context, it's a list of dream properties. It's resistant to enzymes that chew up other proteins. It can handle harsh chemicals and extreme heat. And maybe most importantly, it's radiation hard. It can take radiation doses that would absolutely fry silicon chips and copper wiring. This makes it pretty much the perfect material for the most extreme environments you can possibly imagine. So here's the crucial point. When you have a material that is this tough, this versatile, and it can build itself, what can you do with it? And the answer seems to be almost anything. This quote from a foundational study back in 2003 just perfectly captures the whole paradigm shift. Think about it. You don't need these huge, complex, expensive fabrication plants. You can just program a scaffold to grow itself into the shape of a wire, and then you just coat it with metal. It is bottom-up manufacturing at its most elegant. And the potential applications, they're just staggering. I mean, think about self-repairing hulls for spacecraft that heal cracks on their own. Or 3D food printers that build the actual texture of meat out of a protein solution. We're talking wireless cancer therapy, where injected fibers can be activated by ultrasound to kill tumors. Even the foundations for a new kind of molecular computer. So you see, this isn't just about a new material. It's about a new foundation for our entire technological world. The source material for this explainer lays out this really bold vision for what it calls an amyloid-based future. A future built using the exact same architectural logic we once only associated with deadly diseases. And it even provides a timeline, starting with industrial-scale production in the next few years, moving on to aerospace and food printing in the 2030s, and then leading to full integration across literally every sector of society by 2060. It's incredibly ambitious, for sure, but it's also a surprisingly logical progression. Of course, all of this sounds absolutely incredible, but it brings up the one question that has to be on everyone's mind right now. Given where we started this conversation, is any of this technology actually safe? And the answer lies in this absolutely critical distinction. Researchers are not using the infectious mammalian prions that cause disease. Let me say that again. They are using the architectural principle from safe, non-infectious sources, like prions found in yeast or from fully synthetic proteins they designed from scratch. They're borrowing the self-assembly mechanism, not the pathology. It's kind of like using the logic of a computer virus to build an antivirus program. You use the mechanism before a completely different and totally beneficial purpose. And so, ultimately, we're left with this really powerful idea. This transformation is already underway. The power of this universal material is being unlocked right now. The real challenge ahead isn't one of discovery, but one of wisdom, of truly understanding the incredible potential of what we now hold in our hands.